know quite a bit about Jim's work. So I will just mention he's a primatologist, anthropologist, with lots of contributions in physical anthropology, and of course in evolutionary quantitative genetics. He is now a department chair of biology at Loyola University and used to be a quantitative geneticist and anatomist, I think, the main anatomist of the St. Louis Washington University School of Medicine. I'm going to tell one anecdote because it's appropriate, I think. I was a postdoc with him in 2006, I think, and it was one of these typical interactions where you're just very excited to learn new things about. I did my baby steps in quantitative genetics with Jim, and you run into his office with a new idea, and you think it's really something, and, and Jim just turns around in his stoic way, listens to you, and you start excited in the middle of the sentence usually, and then shows to his drawers where all of his publications were in, and said, 20 years ago, I published that. That was a typical one. So with no further ado, Jim, welcome.
Thank you very much, Mia. I really appreciate the invitation to come here for the week and join myself so far. The weather's been fantastic. But no, it's great to be back in Norway. It's my second trip here. And I also gave a talk last time I was here, probably 2006. So, yeah. All right, so today I'm going to talk about some more recent work and then some totally made up stuff that I'm not sure what to do with, and we'll appreciate any opinions you would have about it. We're going to talk about the evolution of the genotype-phenotype map, and especially with regards to the individuation or parcellation of phenotypes into modules and how that might occur. And there are lots of examples in the world of situations where there's been a break of the tight integration between parts. So the length of the fingers is all very highly correlated in most any mammal. And here we have the ii, who somehow has managed to separate out its third and fourth digits and make them really super long compared to their relative to lemur below. You can also see that the fourth digit's fat and the third digit's hardly there. So they probably are stealing each other's mesenchyme before differentiating. But they use this to tap on the wood like a woodpecker would use their beak. And they tap to find out how to stop, dig in, and eat the insects. They live in Madagascar. So that's a primate example. The integration of the forelimb and hind limb is something we've worked on a lot. And those correlations tend to be very high across mammals. So if you have a relatively long arm, you're going to have a relatively long leg as well. And the question then comes, correlations of above 0.8. The question then comes in, how do you get a bat with extremely long humerus and radius there, and compared to their femur and tibia down below? Those have become unlinked. And of course, there's an adaptation that's involved there in terms of flight. We've seen the individuation of vertebrates. You can tell I really recognized exactly what it was, that it was a lizard tooth row. But you can see the teeth are relatively undifferentiated. They're all the same kind of spike. Whereas in humans, we have the distinction between incisors, canines, premolars, and molars, which is also a common mammalian system. And finally, the individuation of trunk vertebra. Again, it's a human vertebral column on the right, And on the left is the Nectaris, which is a salamander. So what was undifferentiated becomes differentiated and functionally distinct. We use the different regions of our vertebral column for different movements. So it's thought, or at least I'm going to treat the primitive state as a universal pleiotropy and universal polygene. And by universal pleiotropy, every gene, the L's, the locuses on the loci on the left, affect every phenotype. And every phenotype is affected by many loci, all tied together nicely. So how might an evolving system of interrelationships break down this universal pleiotropy and individuate the phenotypes, thereby allowing independent evolution of the traits? So one example, I never talked about this before, so I'll be corrected. This is a work we did on the skulls of the gray short-tailed possum. And you can see there's Arthur. This is part of Arthur's thesis. And the other guy there is Gabriel Marlish, who was also deeply involved in the project. He was Arthur's master's supervisor down in Sao Paulo. These are beautiful little animals. They're monodelphous because they only have one womb. You don't see any pouch for the young. And a whole series of measurements. We have a standard set of landmarks that we take on most any mammal we can find them on. And then look at linear distances between them. So the quantitative genetics of this group is that the heritability was averaged 52%. This is a broad sense heritability because it's based on different partially inbred strains. So 52% is perfectly normal heritability data for morphology in males. But the genetic correlations and the squared correlations are quite high. The average R-square is 0.29, and more than 60% of the variance is accounted for by the first principal component. And that's a general feature of marsupials. I think Arthur worked out in another paper that that's due to differences in life history, that the animals who are born very small and young tend to have everything extremely highly correlated. Not only in the skull, but that's true skull with the pelvis and scapula and long bones, and it's all of a piece. It's not modular at all until you remove size. So we were able to get an F2 intercross amongst these monodelphous strains. There are two strains that were crossed, the atherosclerosis high strain and the atherosclerosis low strain. The guy who generated the lines studies atherosclerosis and the genetics of cholesterol. So, but you know, they have skulls too, just blood, so we figured we could use them. And they had done this intercross between two partially inbred lines. You can see the inbreeding coefficients within lines there. If they try to go to one, they die out. The strains can't be maintained. The coefficient of kinship that's between the strains was 0.24. So we had 576 animals that were measured. 12 of them are parentals, 158 are F1s, and 406 are F2s. And you have to be, we could use the normal analysis that I'm used to in an F2 because of the variability within each strain and the shared alleles between the strains. So there will be gene frequencies that are not going to be 0 or 1. And Arthur managed to do this genotype by sequencing, where we got 3,700 high-quality SNPs, one every 0.25 centimorgans across the 8 chromosomes. The whole genome in monodelphis is 890 centimorgans, so it's a lot smaller, a little bit more than half what a mouse would have. And Arthur found lots of QTLs, found 24 of them. The acronyms for the traits are wonderful. I know exactly what each one of them means. My guess is that it's not going to matter to you guys. The key thing to look at here is the pleiotropy. Now, I found when you do the QTL mapping, you have a significant location that you can't just use the extreme threshold to decide whether a trait is affected at all. When you're doing pleiotropy, you don't want to, you want to allow more traits in if they are affected. And so we use a 5% pairwise threshold, and you can see there are a whole bunch of the degree of pleiotropy is very high. So most loci affect lots of the traits. And there is not a modular pattern to them here. So the correlation matrix that we got from our QTLs there was very similar to those obtained from the quantitative genetic analysis with a matrix correlation of 0.88. The effects are not modular unless you remove size. And if you remove size, then you can get the modules that we're used to seeing in the skull. QTL interval lengths vary from 0.4 megabases to nearly 50 megabases. It's an average of about 12. And the standardized additive values average 0.24 units, and standardized dominance values average 0.2 standard deviation units. And those are the same things we get in the mice, same result. So Arthur compared the degree of pleiotropy in these monodelphins to what we find in mice. Mice are a more normal mammal than monodelphins because, and in most eutherian mammals, things are not all highly correlated all across the skull. Instead, there are modules, or close to having modules. And it took a special kind of test because I'll talk about the mouse data in a minute, but that's in a much later generation. So the QTLs are smaller in a later generation than they are in the earlier generation. So Arthur corrected for that difference and counted up the degree of pleiotropy. And for monodelphis, it's 625, but only about 4.75 in the mice. So there's considerably more pleiotropy. The traits are more tightly tied together and cannot evolve independently like they can in a mouse or a non-human primate, which is what we did most of our work on. So in our mouse population, we wanted to do gene mapping, and the experiment was to take two strains. We were studying growth. I thought the SMJ, small, would have different growth than the LGJ, large. And sure enough, they do. You cross them, get an F1 that's heterozygous for every polymorphism that's different between the two strains. There are only about 6 million of them from the sequencing. So you cross the F1s to themselves, that's why it's an intercross, and you have the F2 animals and the genes segregate out, and the phenotype segregate as well. It's the correlation between the segregation of the phenotype and genotype that you're using to do the mapping. But intervals in these early generations are really big. So the typical interval in an F2 in a mouse would have 400 genes in it. So trying to decide which of those, or what combination of them are causing the effect is difficult. So we started randomly mating them. And when you randomly mate over the generations, you keep accumulating recombination, giving you a finer and finer scale map of the genome. So what starts out as being, what starts out as being 100 centimorgan or 1 centimorgan will become 20 centimorgans. So you stretch the genome out. That way the regions of interest are much smaller and hold many fewer genes. Rule of thumb is that the F34 would have 17 times the amount of recombination as the F2. The F66, which is the ones who are currently alive, that would be 33 times the amount of recombination. But we are getting to the region where, where genetic drift is having more of an impact than it did earlier on. We tried to keep an effective size of 300, but ended up functionally getting 250. So there's just the statistics on that 17 times. We had 3,000 SNP markers every 5 centimorgans along the genome. And the skulls were micro CT scanned and 3D coordinates, anatomical landmarks were recorded. And here's just the drawing of those measurements on a mouse skull. Basically what we have here is the height, width, and length of the brain case and the face, anteriorly and posteriorly, and also the height. Do you notice the bottom picture there, F's facial height anterior is really a short measurement. So it's susceptible to error. Its repeatability is not what the others are. So the heritabilities in the mice, the traits range from 61% to 94%. So that's a lot more heritability than the possums had. And that's because these two strains differed in their size dramatically, while the two strains of Monodelphis did not. So the fact that it's such a big distinction in size is what gives it that high heritability. The average genetic correlation is 0.4, and the squared correlation is 0.19 instead of 0.3. So it's a much lower amount of integration connection between the traits. And again, the genetic and phenotypic correlations are highly concordant with the values we get from the QTLs. So we're doing a good job of reproducing a pattern of relationships amongst the traits from the QTLs themselves. Again, they kind of fail the modularity test as well, in part because size is such a big factor here. And size is going to have the same effect on every trait, so make it longer. And the dominance of size in the cross is what masks the modularity. So if you correct for size, and I took out the first PC, it explains 46.5% of the variance. I think in the marsupials, it's about 60% or 70% of the variance explained by the first PC. These are, both of them are incredibly high numbers. In the non-human primates, it would be more like 30% for the first PC. When you remove size, the remaining correlations average a minus 0.08 and versus a 0.40 when you have size in the traits. And the R squared values go from, comes to as 0.06, so very low integration compared to when size is included. The major anatomical pattern that shows up in this data is a contrast between facial lengths and cranial base and neurocranial widths. So the width of the brain case and the length of the face are tied. And it leads to a negative correlation. So within those groups, the facial lengths they're highly correlated. The neurocranial widths are highly correlated, and the correlations across the contrast are negative. So minus 0.34, and I just did a funny thing with the mouse skull there at the bottom to show it's the wide neurocranium with the short face and the narrow neurocranium with the wide face. This is actually mimics a study I did back in the ancient days, late 80s, early 90s, on artificially deformed human crania. So people around the world, different times and places, have put boards on their head, on their kids' heads after they're born, because they don't want to grow up and have them round-headed. I mean, Germanic peoples are round-headed, but it's obviously inferior to having a deformed, a modified head. So if you're one of these groups used seaweed, wet seaweed, tied it on the kid's head and let it dry. And if you had a round head, it's because your mother didn't care what you looked like, is what the ethnologists recorded. They show the exact same thing. The one with the boards in the front and back end up with a very wide brain case, and their face is short and pulled back in. Well, but the ones with the circle around them are going to have a long, narrow brain case, and they end up with a long, narrow face as well. So it's if you widen the brain case, it's going to pull the face in. If you narrow it, it's going to push the face out. And that's showing up here too. We're still nervous about this because we found 85 loci. A lot. Average of, there were 4 to 5 QTLs per chromosome. Traits had 8 QTLs each. You expect 1 by chance. So some of the QTLs are false positives. I don't mind it. Few of them are. The facial height anterior only had 2 QTLs, and it's likely because it doesn't have a very strong repeatability. So if we consider at these locations, all traits at these locations significant at the 0.05 pointwise level, traits have 30 QTLs instead of 8. So a lot of pleiotropy and polygeny going on here. We calculated the additive and dominance covariance matrices from the QTL effects, and they give pretty good approximations to the genetic and phenotypic correlation matrices we got in the quantitative genetic analysis of this population. So again, we're capturing the relationships. We also, for the first time, looked at clustering of the QTL effects themselves, and we found that QTLs cluster. They do cluster in specific subregions of the total morphological space, but it's not a brain case versus space distinction. The clusters form an unbalanced pectinate shape rather than a balanced bifurcating shape. And there are many QTLs crossing the face brain case boundary. So pleiotropy is modular, it's just not face brain case modular. And only rarely is it antagonistic, where you get a positive on one trait and negative on another. The signs are almost always the same. 16% of the QTLs out of the 85 represented overall cranial size. Everything on is highly loaded. And it's really, those are size measures. 47% of the loci represent some aspect of local size. Not overall cranial size or body size, but local size. And 22 of them represent a contrast between cranial traits. So we mapped long bones before, and Mihaela, actually, I think you might have measured some long bones at one point. Yeah, wow. And when we did the work on the skull, and I was looking through the positional candidate genes, I thought, it just sounds awfully familiar. And it turns out there are many of the same genes. So many of the genes we mapped here for this cranial morphology also affect the length of the long bones. And this is because the growth of both the cranial base and the long bones occurs through a growth plate. There's a common structure, common molecular activity, and that's resulting in the same QTL showing up in both. The long bone is on the left here, and you have a resting region, a proliferative region where the cells are all lined up as they divide and stay in a line. and then the hypertrophic region at the bottom, which then disintegrates in the red as well. So the two there for the cranium are two of these back to back. So resting's in the middle, then you have on either side prolificative, and then on either side the hypermorphic and ossification. So I figured I had to do a statistical test or people wouldn't believe me. So I took my QTLs from the long bones and I just spread them randomly through the genome and saw how often it intersected with the skull QTL. And you can see that typically there was one that was in common, and occasionally two. But the observed was 16. So I think we beat the probabilities there. Now, some places I give talks like this, what they really want to know is what are the genes that are in the QTLs. So here's a list of genes in the QTLs. They have wonderful names. Many of them are well-known bone growth and morphology genes. Many transcription factors. As we're talking about insulin in these mammals, it's the IGF-1, the insulin-like protein 1 that's serves that role all through the body. And we have that. We have its receptor. Just the and the runts are also very important genes. So we got lots of good candidate genes laying around. You go into one of these intervals, and we have the whole genome sequence of both strains. So there's some 5 million, 6 million SNPs, which means in any given region, there's way too many SNPs. You have to start narrowing them down. There are very few protein coding differences between the strains. Almost everything is in the introns or between the genes. And a few of those intronic SNPs are conserved across vertebrates or across mammals, depending on what you want to do. And those are the target ones that we would work with. So how does a modular pattern arise in evolution? We saw there the marsupial, everything is so tightly tied together, you can't break things apart very well, and the mouse less so. So it arises from a state where of universal pleiotropy and polygeny by parcellation. I presume that word actually makes more sense in German, because that's what Gunter Wagner was calling it. And Gunter kind of half translates a lot of stuff when he uses them. So the parcellation is the active process of dividing a unitary whole into distinct parts. And then those parts are free to evolve independently.
This is what they're doing original. You can use it, yeah, but
Yeah. So when we get a parcel, there's a parcel of land would be a field.
Yeah.
So the mail is called parcel. Parcel post is packages that are sent through the mail. So that's what I think of it. You also then would have integration, which is the act or process of uniting distinct parts into a whole. And this can happen through the evolution of pleiotropy. So this isn't the evolution of gene frequencies at the target locus. Those gene frequencies are not involved. Instead, with epistasis, you can have variation in whether the locus is pleiotropic or not, and the extent to which it's pleiotropic. So epistasis occurs when the phenotypic effects of a gene depend on the genotypes present at other loci. The nice quantitative genetic version of epistasis. And this is just this is on fat pad weights, but just to show you an example of epistasis. The D18 MIT51 was a major QTL. The you can see that the column 3 with the blue, red, and yellow is just so much higher than the other ones. So small is dominant to large, and it's a big effect. The proximal part of the chromosome, D18, MIT 17, did not have any effect on the phenotype, because only a quarter of the population, those with a large genotype in the latter part of the chromosome, has an effect, and the other 3/4 don't. If you look at the rows, you'll see that 2 of them have a big effect and 1 doesn't. So 75% of the population had a big effect and 25% didn't. So you flip those two, and you see why you get a QTL in one place and not in another. So pleiotropy can be genetically variable when you have differential epistasis. This means that the epistatic pattern for the same pair of loci are different depending on what traits you're studying. You get a different result for different traits. And with differential epistasis, the pleiotropic effects of a locus are genetically variable and can evolve. So this is the evolution of pleiotropy, not the evolution of correlation. This is just a pretend example of differential epistasis for 2 traits, X and Y. There's no epistasis for trait X at all. It's just regular additive across loci. But trait Y has got all sorts of strange things going on. The capital A genotype is the smallest if you're carrying a capital B homozygote in the other locus. And but if you're carrying the small b homozygote genotype, then the capital AA is the largest. So it will switch depending on which trait you're looking at. So to get an idea of how this might work, the differential epistasis, a modifier locus, suppresses variance for one character, but not for the other at the target locus. And that's just shown here where we have the capital A on the left, and that RQTL genotype blocks the target, the gene of interest from having an effect on trait 2. Whereas if you have if you're heterozygous at that locus, well then the gene will affect both traits. I don't like using modifier and target because whenever you have epistasis, the flip is always true. If A is locus 1 is modifying locus 2 is modifying locus 1. There's no way to break those apart. So we first between the genotype and the size phenotype. Doing this, we found 23 QTLs on 13 chromosomes. 30% of them were in the same location as QTLs that affected the mean, and 70% of them weren't. There was no difference in the mean, but there was difference in the distribution. All traits are affected by interaction QTLs. Superior condylar length, mandibular height, and superior incisor alveolar length being involved in over 5 interactions each. Traits affected by individual relationship QTLs tend to be restricted developmentally to distinct mandibular regions, either the ascending ramus there on the right of the mandible, that's where all the muscles go and attach, versus the alveolus, which is the bone supporting the teeth. Those have very different effects on the growth of those regions. So the RQTLs tended to be restricted to developmentally distinct mandibular regions, either the muscular process or the alveolus. So we found them all over. The red indicates where there's a gene, a QTL that affects the mean and the relationship between traits, while the blue ones just affect the relationship. So here's an example of these. So you can think of this as variation in allometry, if you like, because that's what it is too. When you're using size as your independent variable, these are just three different examples where we found a difference in the slopes of the relationship between a trait and mandible length. Change depending on which genotype was present at the RQTL locus. Right, so the first one up there at the top left, it's the large homozygotes, the solid line. The dashed line is the heterozygote, and the dotted line is the small homozygote. So tell me, does this gene make for a longer posterior corpus height? The answer is, I don't know. The answer is it depends. If you're at a short mandible, the small homozygote has the biggest height. If you have a long mandible, it's the large homozygote that has that effect. So they switch depending on where this interaction is occurring. And you can see this one switches just about in the middle of the distributions. The axes are set up to cover the distribution of the range of the traits and no further. So almost all the time when we found this kind of interaction, we found it occurring with a cross within the distribution of the data. The second one is quite similar to the first. The third one's a little different because it's, for one thing, it doesn't cross in the middle. It crosses on one end or the other. So this one could be a QTL for mandible length at this location, because they're not crossing at the means, they're crossing to the right of the means. So these kinds of variations are variations in pleiotropy, the magnitude of pleiotropy. There's Mihaela. This is a modern picture of Mihaela. I didn't dare use the old one. And this is one of the things you came in and talked to me about, or I came into your office and we talked about it, and you were going, huh, what, where? And that's entirely your reaction to what I'm doing. What's going on here? But Mihaela finally took it a lot further. And this is for different long bone lengths versus body size. So body weight in grams versus the length in millimeters of each of the individual long bones. And you can see that looks just like the mandibular ones. They're crossing in the middle. And you found a whole ton of these too. And then, not being as lazy as I am, Mihaela then mapped the epistatic loci that interact with the RQTL to produce the result. So you can also go and map across the genome for locations that have an epistatic interaction with your RQTL. And that's going to affect these covariances. And you can see again here, this is actually the same graph turned two different ways, right? So you can see that the differences between the genotypes for the phenotype are massively different depending on your alocus genotype. Big difference if you're small a. Not so big a difference if you're heterozygous or capital A. And if you focus on the BB genotype, you can see that you have the opposite direction. On the small b, you have the that's the small homozygous being low compared to the other two. If you look at the capital B, it's reversed. And I could show a whole lot of these. I love these diagrams. But you can see it basically all over. So gene effects on specific phenotypic traits are often context dependent in that they vary depending on the value of an interacting variable, some other phenotype. The range of traits affected by a pleiotropic locus is therefore subject to genetic variation. And it's this variation at these RQTL loci that are responsible. So genetic variation in pleiotropy is due to differential epistasis. And this is where things sat for a long time. I had a nice hypothesis, and I just made up that it was true. Modular pleiotropic patterns are sculpted by directional selection, so that co-selected traits come under the control of a common set of module-specific genes, while traits under independent selection come under the control of separate sets of genes. It took me, well, this was the genesis. Do you recognize the artist? No? Too young. Van Morrison, brown-eyed girl. I went to a concert in England when I was there. I showed up for the concert an hour ahead of time because Americans say the concert starts at the time of 7. That's when the concert starts. In England, it's when the doors open. So I had to go up and have some gin and tonics, and I had this. I had an envelope in my pocket, and I worked out the whole theory there.
That's right.
The other side of the envelope had a whole other one, different thing, but that didn't pan out. But this one seemed to work okay. And basically, we have the RQTLs, where the correlations between two traits vary depending on genotype. And it's going to be this variation we're going to look at. So if we have selection, in this case for a smaller X and a larger Y, we can look at that in a distribution just by changing the axis. So here the x-axis is x minus y instead of x and y. And you can see that on that axis, they have different variances for the different genotypes. Actually, the same thing to be different variances or be different covariances. Just depends on how you orient your traits. And you can see that the small a here has a low variance, and the capital A has a high variance. So that is heterozygote in between. If you then impose a nonlinear fitness function, where in this graph, the A allele is going to be favored because the fitness of capital A is going to be bigger than the fitness of the heterozygote, which is then bigger than the fitness of the small a homozygote. So these variance differences will produce a change in allele frequency at the loci. And I thought of it originally as a truncation selection, which is shown on this slide. And it's easier to see that a much larger proportion of the capital A's are over to the right of the threshold than of the small a's. So the capital A allele is going to be favored when you're selecting in that direction. And it actually can work out in any direction. And the correlation will start at some, make sure I get the right side. The E is showing it starting on at an intermediate value and going down to 0 through this evolutionary process, along with changes in allele frequencies at the RQTL. Or if we start at 0, we can go up to 1, or a very high correlation. Someone's able to go both directions. I think this might be the only way that's been figured out that you can modify pleiotropy itself rather than correlation. And that required a model. So that has a long history too. After I was drinking gin, and the concert was great, I went to, I'd run into Gunter and often Mihaela too at different events. And I'd say, Gunter, figure this out.
It was in Vienna, in now my department.
Yes, it was. That's when I finally bothered him enough, bugged him long enough. We were both staying in the same hotel, even though we were at different meetings, I think. And he walked down one morning and said, "Oh, I got it all figured out." And he thought a bit while he was asleep. Now, I know Gunter thinks in equations. I didn't know he dreamed in equations as well. But he basically mapped out the problem. Mihaela then did the theory to make it come out and different simulations to show how it would operate. And again, here, this is actually haplotype model, so you only have one allele, because having two alleles is harder. Okay. So owing to the different effects of allele substitution, AI for AJ, on trait Z2, different values from trait Z1, the regression slopes between the two traits differs between the genotypes, and that's shown at the bottom part of the figure. And the result was that directional selection results in the integration of traits that are selected together. So the two traits are selected together. They will build a positive correlation. And actually the variance in the direction of selection is increased. The heritable variance is increased. So here you have new variation being drawn out because of the epistatic interactions. So as you can see in that bottom graph C, it starts out with a negative correlation between Z1 and Z2. And then as directional selection going to the upper right is continuing, the distribution rotates so that the main axis of the distribution lines up with the direction of selection. So I consider this real non-Darwinian, and that's not supposed to really happen. It happens when there's this hidden epistatic variation. So likewise, if two traits are selected in opposite directions, one positive, one negative, you'll develop your originally positive correlation will disappear, you'll end up with a negative correlation. Or if you have one trait that's being selected and the other isn't, that pleiotropic link between the two traits is broken. So that you have a lack of correlation between traits that are not co-selected. This just shows some of the numerical iterations that Mihaela did with sustained directional selection. The AI is a high correlation genotype, AJ is low. And you can see that the evolution, it works out fine. You go fixed to the allele with the direction of selection matching the variation within the genotype. So this is Van Morrison effect. In Darwin's theory of evolution by natural selection, new variation is random with respect to adaptive need. That's the usual take on it. And while that's still true here, there's this hidden standing variation that can actually make variation increase when there's an adaptive need. The new variation is, well, it's usually thought of as arising by mutation, but this epistatic variation can become apparent and heritable with changes in gene frequencies at the interacting loci. So the effects of genes evolve under these circumstances. So it's not the change in the effect that you would see when you change the gene frequency at that locus, but the actual effect of the gene itself. The genotypic values rather than the genic values. So the effects of genes evolve, and both the magnitudes of the effects on individual characters and the range of pleiotropic effects will change. So importantly, multivariate directional selection results in increased genetic variation along the selected dimension and an increased rate of adaptation. This new variation arises from transformation of non-additive genetic variation into heritable additive genetic variation by changes of the allele frequencies at the RQTL locus. This new variation aligns with the selected dimension, enhancing the response to selection. This brought me into another area altogether, and this is the part that I just was making up as along. This is a work I've been doing with Benedict Hallgrimsson and Ralph Marcuscio and their postdocs and students on the developmental functions related to morphology of the embryonic face. So the experiments consisted of wild type loci, or where you had sonic hedgehog expression could be artificially increased, and then you'd get a decrease in sonic hedgehog with various knockouts, heterozygous knockout and homozygous knockouts, that produced this logistic curve. Right to the data, and you can see the morphological difference depending on sonic hedgehog expression at the bottom along the x-axis. This is sonic hedgehog that's produced by the brain, the forebrain, and that then affects the width of the face. So you can see that over in the cartoons on the left. You're just looking at just the facial primordial. So you can see it's very much fits this logistic model. It occurred to me though that and you can see, given a logistic curve, that when you're up at the high end of hedgehog expression, there's very little phenotypic variance associated with variation in gene expression. That's what the little red bar over on the left by the y-axis is indicating. The same is true at the low end. So low phenotypic variance when sonic hedgehog expression is low. But there's a high phenotypic variance when you're in the rising portion of the logistic curve. And this fits with their way of thinking of genotypes, because they like to think of genotypes that are important, like sonic hedgehog. And certainly if you knock them out, you get a result like we saw. This higher variation in a heterozygote is common in human syndromes, which are functionally a knockout allele. The heterozygote is a much higher variance than the wild type, as they would say. As long as you're up there in the wild type, though, at the very top of the curve, sonic hedgehog expression doesn't affect the phenotype at all. So we think it should affect the phenotype, but it depends on where your population is sitting on this curve as to whether it does or not.
Wanted to say that has a lot of implications of how we think about comparative studies of gene expression.
It does. And natural variation versus knockout variation. It makes sense to do the knockout variation because you're trying to expand the phenotypic range through that mechanism. So I got looking at this and said, well, sure, if we're wild type, we're at the top of this and we don't show any phenotypic variation during sonic hedgehog. That's fine, but why is that part of the curve flat? While the other part of the curve is steep. Didn't seem right to me that would be some kind of fixed requirement. So I was trying to think of, I've been trying to think of ways to evolve the shape of the curve itself. And by evolving the shape of the curve, changed the steep portion into a flat portion, in which the phenotype is going to be robust to any changes in sonic hedgehog expression. Change sonic hedgehog any way you want, it's not going to matter. And the first thing I thought of, kind of obvious, was this idea, where you could put stabilizing selection on the phenotype. No selection on gene expression at all, no direct selection on gene expression, only on the phenotype. And the original blue curve, I saw this flattening out over time, over evolutionary time, with the stabilizing selection. And that does work to some extent. Unfortunately, when you study this using variation QTLs, which is the one trait version of the RQTL, and if you have stabilizing selection, you will change the shape of that curve. You bring the top down and the bottom up. Unfortunately, it turned out that when we've done BQTLs now, we found that the high variance genotype is the, I'm sorry, the low variance genotype is the heterozygote. So before we were fixing the capital A or small a, here the stabilizing selection would produce a balancing selection on the genotype. And you would have a continuing segregation of the alleles. And they reach some equilibrium value depending on the relative value of the top and bottom of the curve. I think that does work, but it also wasn't, it was limited too, especially with our heterozygotes, everybody being wild type. All of our variant alleles are considered wild type. You don't show the same thing as the knockouts do. So this is a variance QTL. You have different genotypes, have different variances for the trait. And if you have stabilizing selection, and this is threshold selection above and below the mean, you'll see there's no difference in means at this A locus, you would get a narrowing of the distribution because it's going to be the weighted average of the three distributions, weighted by the frequency of the genotypes. So it would, if it was capital A that was the green line, then we go towards fixation of the A allele, capital A allele. So the developmental landscape will have changed while the allele frequencies reach an equilibrium based on relative variances of the homozygotes. So we would get this kind of an effect with the curve flattening. But that didn't seem true enough. So I went to, of course, a variance QTL is an RQTL, they're the same thing again. So we mapped a bunch of variance QTLs for cranial traits in the mice. And we found a fair number of them. There's some 30 of those loci around the genome that affect the variances of different traits. And like the RQTL, these differences in variance by genotype are caused by epistatic interactions with other loci. And these graphs show the epistatic interactions between the BQTL locus and the other loci, the epistatic locus, which is it's over on the right of each of the graphs. Gives you the legend. And here, you're just looking at them, you can see here that in the top left, the variance is much higher for the capital A, and within the small a than it is in the heterozygotes. So in this case, the variance of the heterozygotes is low. So, and you can't fix the heterozygote, but you produce balancing selection in the heterozygote. That's going to maintain genetic variation in your population. So when you go and do a short-term selection experiment, you'll get a response. Also, by the way, peso is weight. And why it's LB, which to me would mean pound, I don't know. No? It's Portuguese. Portuguese post.com, so all the rest is Portuguese. And likewise, the one below it, you can see the capital A genotype has a massive variance, and the others are lower. And on the upper right, you have low variance for the small a, intermediate for the capital A, small a, and large for the capital A, capital A. So by looking at these interactions between loci and how they affect variances and covariances, we can come to understand how pleiotropic effects can change, can evolve. What I've shown in the experiments is that there's genetic variation in the pleiotropic effects. Some genotypes have stronger pleiotropy than others. That's a plain result there. And then the model suggested that, that the selection would produce the results that I talked about earlier. And unfortunately, that's never been tested. I tried to get postdocs and students to do the things that I'm too lazy to do. And one of them is a selection experiment on this, but I thought it would be antibiotic resistance in yeast would be the thing to do, rather than mouse longbow links, which is a much harder selection experiment.
We started. We
Started, went over to Justin Fay's lab, and learned about yeast.
And the bacterial infection of it.
But now you can have a postdoc or a student do it.
Right.
I also thought that the RQTL result might work here as well. So here we focused only on the middle part of the logistic curve. So I reduced the axes down so they're only covering that logistic part, which is a straight line positive correlation. And if with an RQTL, and you select directionally for an increase in the phenotype, since you're not selecting directly on the gene expression at all, you're going to change the covariance in the population from a high positive one to a null covariance. And that's represented by a flat line. So this region of strong relationship between gene expression and phenotype can be eliminated and free the phenotype from the gene expression requirements using this method. So you can see there is a correlated response to selection because there's a positive correlation between phenotype and gene expression. But that positive correlation is broken down and eliminated by selecting on the phenotype, directionally on the phenotype. So adaptation would result in this change in relationship. So RQTL selection can explain patterns of correlation. Select on traits together in the same direction leads to positive correlation, and in the opposite directions it leads to a negative correlation. Selection on only one of the characters selects for a decoupling of the two characters. Like I showed the decoupling there of the sonic hedgehog gene expression level and the phenotype. So this RQTL selection works under sustained directional selection. These are situations where the means are going to be changing. Stabilizing selection, no change in the mean. Under this directional selection, you have adaptation going on, and it's the adaptation that's causing the change in the relationships between the traits. Fluctuating selection, positive, negative, has the same effect. So I call these the results of the Cheverud school. That's my name. Mihaela and I played a game for a while where she would modify pictures I took. So there's a Cheverud Strasse in Vienna, and there's my name was up on the Eiffel Tower. There's, some might think it was just close, but no, if you look, actually, it's there. Well, this one we didn't even have to Photoshop. This one's up there just south of Hamar, north of Stein. That is where it is.
That's where your family's from?
That's where my father's family originally came from, and why we have the name Cheverud that was taken up at the end of the 19th century. I guess the Swedes required it, that you had to have a family name. You couldn't have this primitive, you know, hey, Ud Hansen, Hans Udsen, you know, going on like that in a circle forever. And I think primogeniture also was disappeared at that time as a loss of the oldest son didn't necessarily inherit all the property. And that and the high inheritance taxes caused the family to sell the farm. But there was many of these farms had schools on them. Many of them had chapels on them, churches on them too. I don't think this one had a church. But there are a lot of Cheveruds around Norway. Some of them who look an awful lot like my family. My grandparents migrated independently around 1905, 1906 to the U.S., to Madison, Wisconsin, which was full of Norwegians, still is full of Norwegians. And in there, everybody knows how to say this name, Cheverud. Simple enough, right? But when my father went into the army in World War II, they kept calling him Cheverud all the time. And he wanted to be American, you know, so he didn't want the funny name, so he changed the spelling so that it's you know, people want me to be Cheverud and be French, but that's not the way it works. It's a hard CH because the KJ sounds like a hard CH. And we've been trying to figure out how to say the name properly. And when my brother and I were here over 10 years ago, we traveled with my second cousins. And we asked each of them, you know, how do you say the name? Each pronounced it differently. So we never got a true story about how to say the name. So if you guys know, and I've been told Cheve means different things. My father always told me it was the left side of the farm. But Cheve is jaw. I don't know why it would be a jaw farm. It's a beautiful farm, great black soil, abuts to one of the king's farms. So it must have been good stuff.
In the beginning of the view, basically that is a small part of an old farm. So usually it's the old parts, like this.
All the other places I've been in Norway, it's either a rock or a very thin layer of not very dark soil. But this area, that's why there's a king's farm. And well, the king found it tomorrow as a treaty post for the inside. So these are some of the people who helped with the work that I talked about here, or did the work that I talked about here. Mihaela, and that's an older picture of Mihaela. You look just the same, just the same. Hunter now looks like an old man now. See, he and I are almost exactly the same age. And that was Arthur's thesis defense, after he successfully defended his thesis. So after that picture. So I'll take any questions you might have.
So before we do the questions, so maybe you should ask if there's somebody listening that wanted to ask questions, because we are badly over time. So I think we should just do that, and then we can go to an official.
But there were? Now there are only one. Now there's only one, but there were three.
So wants to ask a question.
Oh, I've asked.
Okay, so minute or two before you can.
Totally unclear, huh?
So any other questions on there, like that one?
Well, thank you for listening.
So, if you think of that curve now at the end, the gene
Logistic.
expression, and you think about physiologically what it means, then usually at least when these genes could be enzymatic reactions or things like that, then there is a theory why it looks like it looks, right?
So these are factors that affect the expression of other genes too.
Right, right. So I was just wondering if you have an intuition what it would mean to flatten it in that sense, because usually you would say that the linear ones would be the very short chains of enzymatic reactions, right?
Well,
And how does it
when you get to
translate into
when you get high at sonic hedgehog, you have a saturation. So
right?
it no longer has an effect on the phenotype
Sure.
because it's got all the sonic hedgehog it can use.
Yeah.
But that's not necessarily a stable state. That's something that can evolve. The shape of the curve.
So it would be, I don't know, the affinity of for the level of gene of the processes, or
How much saturates.
right.
But I literally made up that picture last night when I was trying to.
Yeah, no, sure, but just as an intuition, what it would do.
But it seems that those kinds of levels and values are arbitrary in the sense that they depend on other factors. FGF8 is affected by sonic hedgehog, the differentiation of neural crest cells into the facial primordia, but that's something that happens by sonic hedgehog. So they're transcription factors. I don't know that the metabolic factors would have quite that same shape.
But if you think about like this linear metabolic pathway, and then okay, so then you have a concave curve, so then the right part would look the same, and then of course you will change the shape of the curve depending on what is the reaction rates of the other steps in the process, because it's only the rate limiting step where you actually get down in the concave part of the curve, and the other steps actually flat, and it will change which of them obviously will have. But depending on the state of the other ones, you will have a change in the curve. Even if this is simplest, there is no pathways. So anything that's connected in reactions with the reaction system will depend on the shape of the curve, depend on
all the others, yeah.
Where there you are, maybe more. But you will actually look experimentally.
So you change the rate limiting step.
Right, essentially. So that it to change these things.
Yeah.
There are no questions.
No questions, okay. Yes, so I have a question. Or should we put something? Yeah, there is one person watching. Maybe. I don't know.
Yeah. I guess we could just cut it.
Yeah. Okay.
Yeah.
Bye bye, one person.